This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode number 303, recorded on January 25th, 2024. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from Ann Arbor, Michigan, Michele Swanson.
Hello, good to be with you.
With your Michigan shirt there. Huh?
Yeah, Michigan. But today I want to give a shout out to my Detroit Lions, who are in the one of the last four teams standing for the national championship in NFL football. So they play this weekend to get a shot at the Super Bowl.
Wow, big football in Michigan, huh?
Yeah, and it's been a long time coming for the Lions. So
yeah, the Lions have never won a Super Bowl.
That's right.
Have they played in one?
No, I think they're one of four teams that have not.
Sounds about right.
Something like that.
Also joining us from Charleston, South Carolina, Michael Schmidt.
Hello, everyone.
Michael, are you a football fan?
Yes.
Really?
I grew up as a Bears fan.
Chicago Bears.
Yes,
wow.
where you go to the game and you freeze to death because the Soldier Field is on the lakefront. Now Soldier Field has sort of raised up its side so you don't get the torturous wind off of the lake. But I still can remember one game I went to in December where my thighs, I think, were like frozen chicken thighs. It was so cold.
Is it worse than Green Bay, which I understand is really cold? Right?
No, it's not worse than Green Bay. Green Bay is by far the worst.
Also an outdoor stadium?
Yes.
Oh, my goodness.
Yes. Another outdoor stadium.
All right. That's it for sports, folks. We have science for you, microbiology, two cool stories, and we'll start with a snippet from Michele.
Yes, and the title of this paper is A Data Compendium Associating the Genomes of 12,289 Mycobacterium tuberculosis isolates with quantitative resistance phenotypes to 13 antibiotics. This was published by the Cryptum Consortium out of the University of Oxford in the UK, and it was published in PLOS Biology in August of 2022. I'd like to start by just talking a bit more about this consortium. Their acronym is cryptic. It stands for Comprehensive Resistance Prediction for Tuberculosis, an international consortium. And indeed, in this paper, they have engaged researchers and healthcare workers from 5 continents, and they acknowledge in their paper colleagues from Islamabad, Pakistan, Solna, Sweden, Valencia, Spain, the SynLab in Germany, and then in the U.S., the Center for Disease Control and Prevention, and also the Wadsworth Center's Applied Genomic Technologies facility, and also their bioinformatics core at the Wadsworth Center. So the goal of this ambitious paper was to achieve sufficiently accurate genetic prediction of resistance to most anti-TB drugs so that we can replace culture-based susceptibility testing for TB with whole genome sequence-based. And to do this, they needed to develop assays that would have a rapid turnaround, could be conducted in the clinic at the point of care. So this consortium has set out with a lofty goal of revolutionizing identification and management of multi-drug resistance TB. They are funded by the MRC Newton Fund, the Wellcome Trust, and the Bill and Melinda Gates Foundation. And this consortium has published 19 papers together over the last 5 years. So, wow, are we lucky to have them working on this problem. They also collaborate with 2 interesting organizations, one called Bash the Bug. It is a nonprofit that's based at the John Radcliffe Hospital, also in Oxford, and they recruit volunteers as citizen scientists who work with the platform called Zooniverse, which is to promote people-powered research. And it's got discussion boards, they recruit volunteers, and they essentially crowdsource classification and scoring of images, videos, or graphics according to rubrics that are provided by the contributors. So in this paper, the Cryptic Consortium set up a system so that anyone could access and analyze the images they use to quantify sensitivity to the 13 different antibiotics. And in fact, in 2022, they developed this work and demonstrated that a crowd of volunteers could reproducibly and accurately determine the MICs for TB for 13 different drugs. Now, to do that, they had to develop a convenient and economical method to measure the MIC of several drugs at once in a clinical sample. So they used 96 well plates and a broth dilution mechanism where each well was preloaded with a predetermined amount of each of the 13 antibiotics. And they found when they used this Zooniverse platform and the Bash the Bug consortium that they could calculate the mean or the median minimum inhibitory concentration of the drug. And if they had 11 to 17 independent calculations, they found that was sufficient to generate a reliable number. So they were able to do the experiments in the lab that I'll describe in a moment, upload the images of the experimental plates to this website, and then Bash the Bug volunteers would go on and understand what the criteria were for scoring the plates, and then upload their data. So when you have enough people doing this, you can account for, the error that we all have in our experiments. But when you've got enough people contributing, just 11 to 12 different independent assessments was sufficient to give them reliability. So this is science unlike anything I have done, and I'm just super impressed by it. So what was their motivation? Until the COVID pandemic, tuberculosis has long been our most deadly infectious disease. Each year, 10 million new cases arise and about 1.4 million deaths each year from tuberculosis. Part of the problem is that many of the isolates are antibiotic resistant. So, for example, in 2019, 500,000 cases were resistant to the go-to drug, which is rifampicin. And another 350,000 cases were resistant to more than one antibiotic. Not only that, but there have also been what we call extensively drug-resistant TB, which are resistant to two of our first-line antibiotics, plus fluoroquinolone, and then at least one of 3 injectable drugs that have been developed. So they use this terminology in the paper, and it's not necessary for us to dwell on it here. But know that the burden of multi-drug resistant TB in the U.S., where I have data, is significant. So it costs around $20,000 to treat each person that has drug-susceptible TB. But if they have a drug-resistant or an extremely drug-resistant case, the cost can go up to $500,000 per patient.
And this treatment is not for the average antibiotic therapy of 5 to 10 days. This antibiotic therapy often goes on for 9 months to a year.
And the $500,000 that I cite here doesn't account for the lost income for that patient during these months that they're disabled or having to be isolated, et cetera. So this is a huge problem. It causes a lot of hardship, of course, and it's also a huge cost burden. So there are some technical challenges that they also aim to address, and that is that while we've developed some DNA-based tests that can more quickly and cheaply determine drug susceptibility faster than the traditional lab culture method, and especially with TB, that's an issue because it can take days, if not, is it a week to form a colony? It's a long time.
It's longer than that. Typically, they plant them, and it takes, if they're lucky, 4 weeks, but sometimes as long as 6 to 8 weeks to actually grow that initial isolate. And then you have to do the next round of experiments where you put them into the micro-dilution plates.
So obviously, this is not good for the patient, but also it's quite laborious. So we want to now develop DNA-based tests. And there have been advances in that. There's some nifty, compact, kind of modular machines that clinics can operate, have them on the bench top, and they are effective in many ways. But there are some limitations. My understanding is that their scoring is binary. It's either sensitive or resistant. They have cutoffs that are based on currently available epidemiological data, but they're not as sensitive as the more classic minimal inhibitory concentration assay that is going to be used in this paper. Did you want to add anything to that, Michael, with your experience?
I was just going to add that, when you're going after DNA, you're not actually measuring whether or not the microbe is actually making the enzymes that will inactivate the antimicrobial. You're simply asking, is the resistome present? And that's the value proposition of a minimum inhibitory concentration assay, because you're effectively asking the organism, one, do you have the gene that can degrade the antimicrobial? And two, is it on? Is it on in that circumstance? Now, granted, it's an artificial circumstance, but we have the last 70 years of data where MIC sensitivities and resistance do indeed track with whether or not the antimicrobial is going to work in the clinical medicine realm.
Wow.
So this is a really important observation. If they can correlate the resistome data to MIC data, and it holds true in the clinic, this is a sort of study that says, okay, stop the presses.
Yeah.
We have a new way of assessing whether or not this drug cocktail that we are going to prescribe for 6 to 9 months is actually good money, rather than throwing good money after an organism that's just going to wave at it.
Right. And getting this person back into back on their feet and in life.
Absolutely.
Yeah.
And if you don't think you're at risk for TB, the United States has a substantial tuberculosis population in those individuals in the U.S. that are incarcerated. And unfortunately, the United States has a large incarcerated population, one of the largest in the world.
Yeah. And then travelers would be the other group in the U.S., people that have traveled to regions where it's pandemic. So this cryptic consortium have people from 23 countries across 5 continents, and they collected more than 12,000 clinical samples of TB. And in Figure 2, you can look at the geographical distribution of where these isolates came. So, for example, South Africa contributed 2,000, India 4,000, China 1,000, et cetera, et cetera. Oh, I should say, when they collected the samples, they intentionally biased their collection method toward capturing as many resistant isolates as they could. So they aimed for each resistant isolate to sequence it, but then to compare it to a susceptible strain that was matched in terms of where it was located and when it was located. So that's the basis of this more than 12,000 isolates we'll be looking at. So their workflow is shown in Figure 1. They do whole genome sequencing on these more than 12,000 isolates, and then set up this minimum inhibitory concentration. So this is a classical approach, but it's got that modern twist of using the Zooniverse platform to crowdsource people scoring images of these plates. So basically, it's a 96-well microdilution plate where you've got different dilution series of each of the antibiotics. You inoculate it with the strain, allow it to culture, and then see which wells have got growth and which ones don't. They simply take a photograph of that, upload it to this Zooniverse website, and then crowdsource people to go in and use the rubric provided to determine and enter the MIC for each of the antibiotics. And as I said, by incorporating quality control along the way, they were able to determine that they only needed 11 to 17 independent scorers to come up with a reliable number. So this is amazing.
And so our biostats friends out there will know that more people that agree, and that variable is often measured as your kappa statistic of how often the observers are in agreement with each other. And that kappa value is so important when you're having many people look at the data set to call a plus, and a minus, a minus. So the kappa statistic is what gives this approach its power, because they have so many individuals looking at the raw data.
So in figure 3A, we can see a bar graph that shows the frequency of resistance to each of the 13 drugs that they tested. So for example, 49% of the clinical isolates in their collection were resistant to isoniazid, and then 39% were resistant to rifampicin. And these are two of our first-line drugs that have been in use a lot. Fortunately, 4 of the new and repurposed drugs, so ones that have not been used all that much and are kind of preserved for clinical demand when you have an antibiotic strain that's resistant to our other first and second line drugs, they saw less than 5% resistance to each of those 4 new and repurposed drugs. They did find 3% of the strains were extremely drug resistant, so resistant to the first line drugs plus fluoroquinolones plus another, and 2 strains in their collection were resistant to all 13 antibiotics tested, which is horrifying. And that resistant pattern differed by country, and they've got that data shown in figure 3C. But then in table 3, they show for each of the 8 first and second line drugs, the particular genetic mutation that conferred resistance and how often they detected resistance. So, for example, if we look at rifampicin, which is, again, a first-line drug, this is one that binds to a subunit of the RNA polymerase that makes mRNA. The subunit is called RPOB. And they find that historically it's known that most rifampicin resistance is conferred by mutations within just an 81 base pair region of this gene. And in fact, that was true with their data set. They found that a change from serine at 450 in RPOB, change to leucine, occurred in 62% of their isolates. More rare, they did have 4 missense mutations in other regions of this domain of RPOB, but they were only like 2 to 11%. Interestingly, among the multi-drug resistant strains, only less than 2% had mutations in RPOB. 5% of the strains that were resistant only to rifampicin actually did not have any mutations in RPOB. So that points out that we don't know everything we thought we knew about resistance to rifampicin, which is absolutely known to bind to RPOB and block transcription. So clearly, over the course of lots of exposure of rifampicin and our treatment of TB to rifampicin, other mechanisms have arisen. So perhaps efflux pumps, which pump out the antibiotic. So that was one kind of take-home lesson from this very large comprehensive data set. So that's valuable not only to illuminate the biology of rifampicin treatment and resistance, but also to recognize limitations of our current rapid DNA-based drug susceptibility tests in clinical practice. So for example, we know that there is a high co-occurrence of resistance to rifampicin with isoniazid, again, 2 of our first-line drugs. And that's shown in figure 4A, where on average 80% of the strains that are resistant to rifampicin are also resistant to isoniazid. And that was really the basis for the current WHO guideline that if you detect rifampicin resistance in an isolate, then do not use isoniazid, which is a different first-line drug, different mechanism of action, affects the cell wall. But the problem is that our current, most commonly used DNA-based susceptibility test doesn't distinguish strains that are resistant only to rifampicin from strains that have additional resistance. And we know now from this study that 1% of the strains were resistant only to rifampicin. So there are patients out there who could be safely treated with isoniazid, which is against the current WHO guideline based on, lumping together all of our epidemiological data rather than looking specifically at the genomes of thousands and thousands of isolates. So there is a lot of data in this paper, and I just want to keep it at a high level and emphasize that this wealth of whole genome sequence data on more than 12,000 isolates of TB from 23 different countries with corresponding quantitative minimum inhibitory concentration data for 13 separate drugs really unites molecular genetics and epidemiology and makes it publicly available. So the authors conclude by highlighting that this is an open access resource that they hope will inform future treatment guidelines and also steer development of improved diagnostics. And some of the goals of this cryptic consortium is to investigate for different drugs how particular genetic mutations impact quantitative changes in the minimal inhibitory concentration, study some genetic determinants of resistance to some of our second line and also our new and repurposed drugs, and also use the data to train machine learning modules to predict resistance from whole genome sequence data. And they close by saying that we urge other researchers to explore and analyze this large data set of M. tuberculosis clinical isolates and hope it will lead to a wave of new and insightful studies that will positively serve the TB community for years to come.
This is really a phenomenal piece of work. And I think what's most remarkable is that we now have over 12,000 complete genomes of MTB out there. These are God-fearing clinical isolates. This is not something that had been hanging around in some investigator's freezer that's been subcultured 55 times, so it's no longer the same wild lion that they isolated, 100 years ago. These are still virulent pathogens. And so we have 12,000 plus sequences of this pathogen that has been afflicting the human race for over 12,000 years.
And it's impressive, too, that they really represent many, many different regions across the globe.
I was especially impressed with South Africa.
Yeah.
I have a colleague in South Africa that worked with me on the copper project, and she was always looking at could copper surfaces introduced to her tuberculosis clinics cut down the incident transmission of TB in that clinic, because many of the patients that she was seeing, as the figure so elegantly shows, were resistant to many, many of the first-line drugs that she had available. And South Africa has a significant TB problem, as you can tell just by looking at those figures, and they're resistant to the drugs that we have to control this. And again, South Africa has very crowded conditions in some of the areas of the country where they still are, trying to address housing issues, and crowding is one of the principal ways TB is, of course, transmitted. Your risk factor, of course, is simply breathing.
I was amazed to see how many isolates from Peru.
Yes.
Yeah.
I didn't realize it was such an issue there.
2,700.
Yeah.
It also tells you that Peru is investing in public health.
Right. It's a credit to their infrastructure that they are participating and being aggressive about collecting information.
Michele, is one intent to someday use genome sequencing to assess resistance and not do MIC anymore?
That's their hope. Yep.
Do you think it is possible?
I'll toss that question to Michael, who has more experience in the clinical microbiology.
I think in the case of tuberculosis, where the organism grows slowly and you don't necessarily have the fulminant infection where you need to get drugs on board, and you make the mistake of empiric prescribing just to save the patient until you get sensitivities back. I think there it will have an impact on these slow growers like MTB and even in the non-tubercular Mycobacteria. We right now have an outbreak in individuals that have lung issues, whether it's bronchiectasis, that they get NTM infections. And these people then need to go on a 3-drug cocktail for a very long period of time. And again, NTM is non-tubercular Mycobacteria, and there's at least 200 different species of Mycobacteria out there, and they're as nasty to treat as TB. Some grow fast, some grow slow.
The other possibility is that with Cryptic generating all this data in time, it may be that we can refine and come up with the next generation of these current machines, like Gene Expert is one of them, to look at more alleles across the genome and to make more accurate their predictions.
Mm-hmm. Yeah. All right. Thank you, Michele. Very exciting. Yeah. Michael, do you have your heart in this one?
I do indeed. I do indeed. Just in time for Valentine's Day. This episode will probably drop somewhere in the vicinity of Valentine's Day. And one of the titles that caught my eye was, "Can our microbiome break our hearts?" And then it had the next clause, "Collaborative production of paracresol sulfate and indoxyl sulfate by commensal microbes increases susceptibility to thrombosis." The title I just quoted is from a commentary that appeared in the January issue of MBio by Machado Rubio, Brito, and Binlos, who are at Vanderbilt University in Tennessee in the Department of Pathology, Micro, and Immuno, and their respective Howard Hughes Medical Institute. And they are commenting on the paper that appeared in the November-December issue entitled "Microbe-derived Uremic Solutes Enhance Thrombosis Potential in the Host." And this is by Nemeth and colleagues. There's quite a few authors, so I'm respect to all of the authors, but for the sake of time, I'm just going to offer that they are from the Department of Cardiovascular and Metabolic Sciences at the Lerner Research Institute, which is effectively the research arm of the Cleveland Clinic, which many of you have heard of, which is located in Cleveland, Ohio.
But there are also collaborators from Stanford, from UC Davis, from Caltech, from Madison, University of Wisconsin-Madison. So, and then the Chan Zuckerberg Biohub in San Francisco. So this, again, is another large team effort.
This is another large team effort. And when we learn about the data, you're going to understand why. So I dropped in the show notes both a copy of the paper as well as a commentary. Depending on how much time you have, you can either read one or the other or both. So there exists a body of data that suggests that metabolites produced by our intestinal gut microbiome can affect cardiovascular disease. And this is the big but, the mechanism as how these metabolites work in influencing our risk for developing cardiovascular disease is largely unknown. Cardiovascular disease remains the leading cause of mortality worldwide. This is despite significant advances in preventative cardiovascular treatments and prevention efforts, everything from taking statins to reduce lipid levels, doing your cardio every day, exercising, losing weight. However, there still remains a significant residual cardiovascular disease risk with numerous atherothrombotic events. And an atherothrombotic event is a progressive disease characterized by the accumulation of lipids, fibrous material, and minerals. Some of us who have reached a certain age have been encouraged to get a calcium CT of our chest to see whether or not our coronary arteries are getting calcified. So all of this rolls into whether or not our microbiome can actually influence it. So this then all rolls into cardiovascular mortality risk occurring even in those optimally controlled individuals. And so I have dropped a review article into the show notes because I'm not a cardiologist. So I put atherosclerosis review into the show notes if you guys are interested in taking a look at it. But our understanding of how metabolism and the end products produced by our gut microbes is something in our wheelhouse. And these authors are going to take us on this fantastic journey describing how these end products produced by our gut can actually impact cardiovascular disease. And they're effectively dropping that gauntlet, effectively calling out that cardiovascular disease risk warrants the attention of all microbiologists. Now, the metabolic end products that we're going to discuss are paracresol sulfate, that they abbreviate PCS, and indoxyl sulfate. These are both gut-derived metabolites that have been traditionally associated with cardiovascular disease risk because when the levels of the paracresol sulfate and indoxyl sulfate accumulate in the bloodstream as some individuals' renal function declines, these compounds are considered uremic toxins because they have been implicated in mediating this adverse phenotype. And the adverse phenotype has generally been seen in the setting of renal disease. In other words, your kidneys aren't as efficient as throwing these toxins out of you through your urine. And their synthesis has been referred to as being meta-organismal, since each is co-synthesized by initial an obligate gut microbiota dependent process, followed then by the host enzymatic transformations that then facilitate this metabolic excretion through, in this case, our kidneys.
Wow.
Each of these molecules begins simply as a byproduct of bacterial fermentation, normal parts of the metabolism of aromatic amino acids from a diet that's rich in protein. So as the Western diet is rich in protein, this could explain why cardiovascular disease has been on the rise in the 20th and now 21st century.
Well, let's check that. Let's do some science and check out that hypothesis, Michael.
Hey, that's what we're going to do, folks. In the case of indoxyl sulfate, tryptophan from the diet can be degraded into indole by the microbial tryptophanase gene, encoded in the genome of a variety of gut commensals. So this isn't a pathogen. This is something that normally lives down there. So once the indole is produced, it is then absorbed via portal circulation, goes to the liver, and then the liver, which oxidizes it via the liver enzyme, CYP2E1, and then it's sulfatated by another host enzyme, SALT1A. This then paracresol sulfate then follows a similar production in its production of making the sulfonated form that then is excreted. So dietary protein in the case of the cresol sulfate is derived from, rather than tryptophan, it's derived from tyrosine. And it's metabolized by our gut microbiome to yield this molecule. The liver physiologists well understand this notion of what a meta-organismal biochemical transformation is. And so into the show notes, I dropped a review from 2011 that was senior authored by Margaret McFall-Nye, where she goes into what a meta-organism is, if you're interested. So that's just something for your enrichment. In the paper by Nemeth, they walk us through how they identified the microbial pathways responsible for producing these uremic toxins, or as they call metabolites, that then adversely impact the host, as they offered that it was only through the identification of the pathways that we will be able to then and only then begin to develop therapeutic interventions. So their interest in these two molecules, PCS and IS, resulted from, and this is the really clever part, from an untargeted metabolic investigation where they discovered and identified the microbial precursors of this PCS candidate molecule, whose circulating blood levels were able to predict the incident cardiovascular disease risk among subjects with not abnormal kidney function, but with normal kidney function.
And Michael, when you say subjects, we're talking about two-legged people. Yes, not mice.
We're talking about human beings.
Yeah, not mice.
We're talking about us.
Yeah.
We're talking about us, folks. And this isn't a study of, 15 people in the lab. This is 1,149 subjects where they drew blood samples, isolated the plasma, and all of these people had relatively good kidney functions. And then through a very sophisticated assay. They linked the blood levels of these defined microbial precursors to cardiovascular disease risk. The title of this section of their paper is Uremic Solutes are associated with overall, underscore this word, mortality in individuals with preserved kidney function. We're killing folks.
You've got my attention.
Their approach involved using an instrument that is quite common in many hospital laboratories, untargeted gas chromatography time-of-flight mass spectrometry, or GCMS-TOF. And they observed a derivatized analyte consistent with the precursor for the PCS. And here's, again, the stop the presses part. The microbiome metabolite and precursor to PCS was noted to be significantly higher among subjects who died over the ensuing period of, they followed these people up for 5 years. We're talking 1,149 people that they followed up for 5 years, wow, compared to people who did not die. And this is in their figure 1B, and the p-value is 0.0001. So the sample size is sufficiently large and the data are sufficiently compelling. But the authors are critical of their own work. They candidly offered that they were surprised that the paracresol finding, because their GC time-of-flight mass spectroscopy method was not optimized for the detection of this volatile compound. And further, the untargeted, or I should maybe say unbiased analysis as they first performed on this very large cohort of plasma samples was not quantitative. It was just a plus or minus. And so given the novelty of their findings, where PCS was previously linked to cardiovascular disease and mortality risk, they then decided to use a more sensitive, quantitative method to validate this observation in, again, non-end-stage renal study subjects. Here, the study subjects are now 3,954.
Wow.
They used a complementary technique. They used stable isotope dilution, LC-MS-MS, and they explain it all beautifully in the materials and methods. And they expanded their look to see, in addition to the PCS, whether indoxyl sulfate, the other molecule associated with increased cardiovascular disease, was present. Now, the liquid MS products that they were looking for was done using, again, this cohort of approximately 4,000. They used this isotope dilution mass spec approach, looking now for the concentration of the precursors, and they were able to associate these uremic toxins with an ability to predict a 5-year mortality risk, and that's figure 2. When I read this and then appreciated the significance of that simple blood test, I was like, wow. And then they went on to slap me again upside my head with even more data. And so here the association between these two risk molecules, if you will, is the incident of death holds true when the subjects were divided into subgroups with relatively preserved kidney function. And here they define what kidney function is. It's the estimated glomerular filtration rate, which, if you are not familiar with GFR, it's a test used to check how well your kidneys are working. And specifically, it estimates how much blood passes through the little filters in our kidney called the glomeruli each minute. And so a blood sample is set to the lab. Then what they measure is the creatinine level in the blood sample. The creatinine is a chemical waste product of creatine. And then a lab specialist combines your blood creatine levels with several other factors like your height, your weight, your age, to estimate your GFR. If your GFR is over 60 mils per minute, and then you're considered okay from kidney function. And if it's under 60 mils per minute per 1.73 meters squared, you're considered to have impaired kidney function. And again, this is a test that many of us get each year when we do our annual physical, and they always look at GFR. It's on my chart value. I looked at it to see where I fell in this category, and it's really something that, those of you who have access to your medical record lab tests, you can take a peek and see where you might fall in this list. In further sensitivity analyses, they observed the association between PCS and IS and mortality risk remained significant in both males and females alike, younger versus older, as well as within many multiple different subgroups, including subjects with or without the high lipid rates or hypertension. These are two predictors of whether or not you're at risk for cardiovascular event. If you have fat lipids or if you have high blood pressure, they're always worrying about whether or not you're going to have a heart attack. So bottom lining this so far, the data convincingly show that these PCS and IS, which up until now were only shown to be relevant for cardiovascular disease in folks with kidney disease, are now associated with mortality in the broader population. Like I said, these authors have, information that the microbiologists really need to say, okay, we need to begin to think about this.
Although right now it's an association, we need to do some experiments, genetic experiments in mice to test it.
So here we go. That's step 2. Michele effectively took us to where the authors are going to go next. We're going to learn how they were first able to identify the microbes and the enzymes responsible for the generation of these uremic toxins, and then using an approach many of us used in the past, simple, straight up bioengineering, making mutants. They engineered human commensals to produce these molecules, the PCS and the IS, and then using gain of function and loss of function mutants, an approach that many of us used routinely in our labs.
That's classic.
They did it both in vitro and then in vitro, colonizing germ-free mice. They followed up by studies they first looked in the mice to see whether or not they had increased thrombotic events, and then they followed it up in people. So what they did first, and again, they had no clue as to what microbes in our complex gut were responsible. So they took advantage of previous work that showed that this enzyme complex, HPD, BCA, and Clostridium difficile, which encodes a multi-component 4-hydroxyphenacylate decarboxylate, which can convert 4-HPA and to a lesser extent tyrosine. Again, we're burning amino acids. That's what they're looking at. They're looking at the enzymes responsible for the burning of the amino acids.
And Michael, these enzymes and particular pathways aren't going to be on the test, right? I don't need to.
No.
Okay.
No.
We can keep it high level. Okay.
You need to know how they did it, because it really goes into what the microbiologists have to begin to think about. And they ultimately found one candidate organism, Blothia hydrogen-trophica, and a Clostridium species called D5. And they had homologs of this enzyme responsible for the conversion of the tyrosine to the PCS. And this was this is NIH's money well spent. These data were found in the reference genomes from the National Institutes of Health Human Microbiome Project database. So your money well spent, folks.
Tax dollars at work.
And Blothia is a genus of anaerobic bacteria with probiotic characteristics. So it's thought to be a good guy that occur widely in feces and intestine of all mammals. And based on the phenotype and phylogenetic analyses, some species in the general Clostridia and Ruminococcus have been reclassified as Blothia. To date, there are about 20 species of Blothia out there. They then tested the capacity of the two strains, Blothia and the Clostridium D5, to produce the PCS from tyrosine and 4-HPA under anaerobic conditions. While both strains were able to produce the PCS robustly from 4-HPA, which is the precursor, and production from tyrosine was negligible from the Blothia. So this is sort of a wrinkle. They were able to make the product from the second product in the chain, but they weren't able to make it from the tyrosine, which starts the train wreck. They weren't able to make it from that. So given that the metabolic byproducts excreted by one bacterium are often utilized by their neighbors in the gut community, they formed this alternative hypothesis, namely that the synthesis of PCS from the tyrosine could be the result of a collaborative pathway involving two microbes.
A food chain.
A food chain
among the microbes in our gut. Yeah.
Exactly. And they performed studies to support this hypothesis that two microbes contribute to the overall tyrosine conversion to PCS within our guts. So here again, a page from old-time religion. They used a co-culture approach. They engineered it into two different species of Bacteroides, Bacteroides theta iota micron and vulgatus. They cold cultured. They fed them tyrosine to the pair, where the first strain was a 4-HPA producing or non-producing Bacteroides, and the second strain was a Clostridium that converted 4-HPA to the PCS. And you get all of this in figure 4. You can witness for yourself the production of robust amounts of the PCS. Next, they said, will this work in an animal? So they next colonized germ-free mice. So they had their two engineered strains. They put it in the germ-free mice. They tested it in vitro for about a week. And as expected, they saw recapitulation of their in vitro data in vivo. They witnessed robust production of PCS that led them to seeing PCS in the urine of the mouse. Because unless you see it, you don't know if it's really working.
That must have been an exciting day in the lab, huh?
I am amazed at this paper. I kept, looking at this and I'm going, this is so cool. The next batch of experiments was an exercise in enzyme engineering where they, again, took these strains. So we have the smoking gun phenotype, which was the development of this prothrombotic fibrin clot that manifests or is visualized as the faster formation of this dense meshwork composed of thinner and highly branched fibers that are relatively resistant to plasmin-induced lysis. Now, this phenotype has been seen in patients not only with myocardial infarction, but in stroke, venothrombotic events, deep vein thrombosis, and pulmonary emboli. And what they then measured in their in vivo animal studies were two parameters: the rate of platelet clotting following carotid artery injury and the time to cessation of flow within the carotid artery. And again, this all rolls into the being able to detect the phenotype and effectively demonstrating a mechanism how these two metabolites are involved in the mechanism of cardiovascular disease.
Yeah, they're microscopic images showing the formation of these thrombi is really disturbing.
It's
my goodness.
It really should put a call to action for microbiologists to begin to think about the microbial ecology of our gut and what our commensals have the opportunity to do if all of a sudden we eat too much protein.
Yeah, and hopefully how our diet contributes, hopefully.
Yeah. So in the third phase, they colonized germs-free mice and they took the engineered strains and demonstrated that the gut microbial genes for PCS and indole tryptophanase were sufficient to confer the prothrombotic phenotype in vivo. So we're still in mice. Now, what about people, folks? Well, the final piece of data that they offer us is that using a human fecal metagenomic approach, their analysis, and this is all laid out beautifully for you in Figure 7, offered that the abundances of the two enzymes, the HPD, BCA, and tryptophanase, were indeed associated with cardiovascular disease from the bugs that were recovered from human feces. So this study showed us that these two things that are normally disposed of out of our urine, if their levels get too high, these two abundant microbiome-derived metabolites play a broader potential role in cardiovascular disease than previous was known. Before, it was known that they were associated with people with impaired kidney function, and they correlated with this. But this one really is showing that, even normal, air quotes, healthy individuals can have these reactive molecules in their bloodstream that if you do have one of the other co-risk factors, like your lipids are too high, you have the ability to lay down calcium in your veins. So in this beautifully laid out paper, they showed a linkage between two microbial products and significant human diseases. And I was convinced through this careful and deliberate approach that we now have our first glimpse into a mechanistic understanding of their biological effects and its relationship to cardiovascular disease. And of course, can we figure out how to reduce this rate or change our diets, or what can we do to lower our risk?
Or
and
measure
that's
it?
where they end.
Measure it.
Well, I would start
people.
with measuring it.
Yeah.
So the implication is that our microbiomes are the same, but people who have cardiac events have other issues that combine with these two compounds, right?
Yes. That they somehow are participating. I'm sure more work is being done. People are trying to engineer microbes. There's some older literature about engineering the microbiome, but I think aromatic amino acid metabolism is ubiquitous in our gut flora. I mean, it's a good source of energy. So we're going to see it. So I don't think we're going to be able to engineer out the catabolism of tryptophan or the catabolism of tyrosine. I think those two are going to be around. And as long as maybe we're going to go to a lower protein diet.
Can you control the levels of PCS and IS, for example?
I don't know, and they didn't get into that. So,
but now
I
that
got
we
to talk
know,
to my friends.
Now that we know those are two molecules we should be paying attention to.
Yeah.
Presumably, somebody can do that work, huh?
I think we need to talk to our friends who are the hepatologists, the folks who understand livers, and maybe we could develop a drug that would prevent the sulfonation. We don't know which molecule. Is it the sulfonated form or the precursor that is responsible for the increased in risk?
Yeah.
And there's probably people out there that have, wickedly clean arteries all the way to their hundreds. And we have to figure out what their genotypes are.
Yep.
I think we're going to have to use a G-Whiz assay in order to figure this out, genome-wide characterization.
Michael, forgive me for asking a detailed question about death. The bar graphs are convincing that it's associated with higher incidence of death within 5 years. Do we know what that translates to, really?
How many people?
Yeah, or how many months will it shave off your life?
I don't know how to do those calculations.
Me either, and I don't see it.
I have to talk to my friends across the street in cardiology and gastroenterology, and I sent these two papers out to all my friends in infectious disease. I told them to have their fellows read it because I think this is a brave new world for infectious diseases because they are the ones who understand what's going on with our microbiome. They're beginning to investigate it. Infectious disease doc, you just think treat infections, but in reality, they're the microbiologists of medicine. And they really understand this, and they know much more in terms of liver physiology, kidney physiology, and so I sent this paper off to my friends in ID, nephrology, and GI to gauge their thoughts on this. And As Elio used to always say, stay tuned.
Yeah.
I'm sure cardiovascular disease costs the Center for Medicare Services a bunch of money each year. So this is something that I think the NIH is going to take up the gauntlet and really begin to investigate. So you microbiologists out there, stay tuned. This is where your next grant might come.
Nice.
Or the call for a grant, the RFA.
Thank you, Michael. Two very impressive studies today. Just microbiology keeps on giving, doesn't it?
Yeah.
It does indeed.
And human health for both of them.
Yeah. All right. That is TWiM 303. The show notes are at microbe.tv/TWiM. If you like our work on these programs, consider supporting us. We'd love to have your assistance financially. Microbe.tv/contribute. Microbe TV is a nonprofit, so you're in the U.S., your donations are federal U.S. tax deductible. If you have any questions or comments, please send them to TWiM at microbe.tv. Michele Swanson is at the University of Michigan. Thank you, Michele.
My pleasure.
And Michael Schmidt at the Medical University of South Carolina. Thank you, Michael.
Thanks, everyone. Have a good month.
Good month. I am Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Microbiology for their support of TWiM, Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.